cDNA libraries. So before I get into the detail कि cDNA libraries क्या है, I would like to tell you about genomic libraries. तो genomic DNA libraries होता है और cDNA libraries होता है. तो genomic DNA में क्या होता है ना? Genomic DNA is a chromosomal DNA of an entity representative of collection of its genomic content. तो genomic DNA library में हम लोग पूरा DNA का कॉपी बना सकते हैं और बाकी भी हम लोग इसका बाद में डिस्कशन करेंगे. तो फॉर इंट्रोडक्शन अबाउट cDNA library is a collection of cloned cDNA sequences, each representing a particular mRNA molecule in a cell or a tissue. It can be used for gene discovery, gene expression, analysis, and other applications in molecular biology research. So how is a cDNA library created? So mRNA is extracted from a cell or tissue sample. Reverse transcription is performed using a reverse transcriptase enzyme and a primer. The resulting cDNA is cloned into a vector and transformed into bacteria or other host organisms. तो इसके steps होते हैं. We have 5 steps. So step number 1 is mRNA isolation from cell or cDNA synthesis using a reverse transcriptase enzyme. इसके बाद हम लोग step 2 cDNA synthesize is joined to T4 ligase and 5' is removed using alkaline phosphatase. So step number 3 is cooperation of this fragments into a suitable vector like plasmid or phage और फिर introduction of recombinant plasmid to a suitable host. जैसे कि बैक्टीरियम हो गया, example E. coli. फिर इसके बाद वही multiplication, screening, selection and characterization of clones होगा. And step number 5 is maintenance of the set of clones petri dish में. So types of cDNA libraries, तो normalized library है, subtractive library and directional library. So normalized library में क्या होता है? Contains equal amount of cDNA from each mRNA sequences species, reducing the representation of highly expressed genes. Subtractive library is created by hybridization cDNA from 2 different samples and subtracting the common sequences, allowing for identification of differentially expressed genes. And directional library क्या होता है? Created with a specific orientation of cDNA fragments, enabling expression in the correct reading frame. तो जैसे मैंने पहले मेंशन किया था कि cDNA लाइब्रेरी होता है और जीनोमिक लाइब्रेरी होता है. तो cDNA में कैसे होता है कि सिर्फ कलेक्शन ऑफ क्लोन, मतलब जो कॉम्प्लीमेंट्री डीएनए रहेगा टू अपना mRNA एंटिटी, सिर्फ वही प्रेजेंट होगा और रिप्रेजेंट्स जीन्स एक्सप्रेस इन अ पर्टिकुलर सेल एट अ गिवन टाइम. और ये जीनोमिक लाइब्रेरी से कंपैरेटिवली स्मॉल होता है इन साइज और इसमें हमारे पास कोडिंग सीक्वेंसेस होते हैं और एंजाइम होगा रिवर्स ट्रांसक्रिप्टेज. Just a second. Okay, so material required to start is mRNA और इसका expression in prokaryotic system is easier as compared to the genomic library क्योंकि इसमें coding sequence ही present होता है. और रिवर्स ट्रांसक्रिप्टेज हम लोग कहाँ यूज करेंगे? सिंथेसिस करेंगे जब cDNA का फर्स्ट cDNA स्ट्रैंड को सिंथेसाइज करेंगे तब. सो एप्लीकेशंस ऑफ cDNA लाइब्रेरीज, जीन डिस्कवरी में यूज होता है, जीन एक्सप्रेशन एनालिसिस में यूज होता है और प्रोटीन एक्सप्रेशन में यूज होता है. तो cDNA libraries can be screened for new genes which can be further characterized and studied. cDNA libraries can be used to compare the expression of genes in different tissues or under different conditions. Protein expression, cDNA can be expressed in a host organism to produce proteins for further study or use in biotechnology. तो जैसे इसका एडवांटेजेस है, इसका डिसएडवांटेजेस भी है. तो डिसएडवांटेजेस या फिर इसको हम लोग लिमिटेशंस बोल सकते हैं. तो रिप्रेजेंटेशन, तो cDNA libraries may not represent the entire transcriptome of a cell or tissue क्योंकि इसमें सिर्फ कोडिंग सीक्वेंसेस दिखेंगे and may not include rare or low expressed genes. The size of cDNA fragment may limit the cloning and expression of some genes and contamination भी हो सकता है. cDNA libraries can be contaminated with genomic DNA or other non-target sequences. So in conclusion, cDNA library is a valuable resource in molecular biology research. It can be used for gene discovery, expression, analysis, and protein production. However, the limitations of cDNA library should be considered when designing experiments and interpreting results. तो हम लोग इसका एप्लीकेशन हम लोग डिस्कवरी ऑफ न्यू जीन्स में कर सकते हैं, या फिर क्लोनिंग ऑफ फुल लेंथ cDNA मॉलिक्यूल करना हो फॉर इन विट्रो स्टडी ऑफ जीन फंक्शन, उसके लिए कर सकते हैं। और mRNA टू स्टडी वेरियस different mRNA expressed in different cells or tissue and study alternative splicing in different cells or tissues. So major disadvantage of cDNA library is that it contains only sequences that are present in a mature mRNA. Introns and any other sequences that are altered after transcription are not present, such as promoters and enhancers. ये sequences भी नहीं transcribed होते हैं, इसकी वजह से ये भी absent रहेंगे in cDNA library. So that's all for my presentation. P32. Probes as in RNA probes. नहीं sir, DNA probe. DNA probe. Yeah, DNA probe. वेक्टर्स में हम लोग बैक्टीरियोफाज यूज कर सकते हैं, प्लाज्मिड यूज कर सकते हैं। इशरेशिया कोलाई, हेलो, पीसीडी वेक्टर। थैंक यू सर।
हाँ सर। यस। So is it visible? So good evening everyone. Myself Siddhi Vijay Kulkarkar and my roll number is 227317. So my topic for today's presentation is screening of cDNA library. So first, before going into the details of screening, we'll see what is a cDNA. So basically, cDNA is a complementary DNA or a synthetic DNA which is transcribed from an mRNA, mature mRNA, by a reaction in which enzyme reverse transcriptase is used. So these are the steps for synthesis of a cDNA. So in the first step, a mRNA molecule, whichever we want, the specific mRNA molecule is used as a template for the production of the DNA strand. So a very short oligo-DT primer is unable to the poly A tail. So as we know that we use a mature mRNA, so which has a poly A tail. So we use a short oligo-DT primer of 12 to 18 nucleotides, which will bind to the poly A tail. And then this oligo-DT primer will act as a our reverse transcriptase will use it as a primer to synthesize the DNA strand. Then RNase H will degrade the RNA fragment, mRNA fragment, and but it only partially degrades it. So therefore, the remaining small fragments of degraded RNA will be used as a primer by DNA polymerase 1 to synthesize the other DNA fragment, and the remaining gaps will be sealed by DNA ligase. So a double-stranded cDNA will be formed. So this is the diagram in which it is explained. So as we can see that there is a poly-A tail to which oligo-TT primer is bind. Then we can see the DNA strand which is produced by the reverse transcriptase enzyme. Then in the next step, RNase has degraded the RNA, which is the green color small fragments are left of the degraded RNA fragments, and then DNA polymerase 1 will construct a new DNA strand, and DNA ligase will ligate the gaps and will get our required double-stranded cDNA. Then what is cDNA library? So cDNA library is basically a collection of clone cDNA sequences which are complementary to our mRNA, which we have extracted from whichever organism or tissue which that specific mRNA. So then cDNAs are cloned into a vector so that they can be transformed into a host cell so that we can preserve them in the expression libraries. So what is screening a cDNA? So basically screening means searching for a cDNA clone which encodes a specific protein, whichever or specific protein or desired protein which we want to study. Then mostly the screening is done by using antibodies which are specific or which can bind to our protein of interest. So now steps in screening cDNA library. So first, a cDNA will be cloned in an expression vector, and then a bacteria, like here, example of E. coli is given. So it will be transformed with that expression vector. So then the cells are plated, and individual colony is transferred into the well of a microtiter dish. So now the pattern in which the colonies are grown on the microtiter dish, the same replica will be taken on the membrane filter, which is placed on a selective medium. Then this filter will be peeled and the cells will be lysed. So the proteins which is within the cells and those expressed by rcDNA, the specific protein, that will also stick to the filter. Then by any probes, in this case there is labeled antibodies, so which are specific to a protein, we'll add them, and we can they will bind to the target protein, and we can check them or we can detect this binding by the protein and our probe by various methods. If the antibody is radioactively labeled, we can check it by autoradiography, or if it is not radioactive labeled, we can check it by fluorescence or various other methods. So now applications of a cDNA library. First of all, a very common application is the storage of information because cDNAs do not have non-coding regions. So in a reduced amount, we can store our information. Then cDNA libraries are also used while reproducing eukaryotic genomes because again the same because they do not have non-coding regions. So we get the exact coding sequence. Then cDNA libraries are also used to express eukaryotic genes in prokaryotes. As we all know that in prokaryotes there are no introns. Introns are absent, and in cDNAs also there are no introns. So we can easily express cDNAs into prokaryotes. Then cDNA libraries are also used in reverse genetics, where additional genomic information is of less use. So in reverse genomics, what we do, we study a function of a particular gene or sequence by its effect, by analyzing its phenotypic or genotypic effect. So here we do not need to study the whole genome of an organism. We need only the exact sequence, and that we can get from cDNA libraries. So another application is cDNA libraries are frequently used in functional cloning to identify genes based on the encoded protein's function. So here we usually screen a cDNA library, and then the our protein of interest which we get after the screening is sequenced and then cloned. So it is very helpful in functional cloning. And then for studying eukaryotic DNA, expression libraries are constructed. So eukaryotic DNA can be easily studied by using cDNA libraries because we know that the cDNA sequence is truly a gene, that it has no non-coding sequence. These are the references. Thank you. Yes, sir. Expression vector consists of a promoter. Hmm. Hmm. Human insulin gene and human insulin DNA. E. coli. एक्सप्रेशन वेक्टर में क्लोन किया तो ना, देन सी डीएनए वाला। सर क्योंकि वहां पे तो प्रमोटर नहीं रहेगा ना, वहां पे एक्सप्रेशन वेक्टर की जगह बट नॉर्मल जी वर्सेस सी डीएनए। तो सर हमारे नॉर्मल जीन में तो लाइक इंट्रॉन्स वगैरह भी रहते हैं ना, की वजह से। हाँ एक लैम्डा जेड डी करके वेक्टर है जिसको यूज किया था स्क्रीनिंग के लिए। एक्सप्रेशन वेक्टर ईस्ट में यूज होता है या नहीं, नहीं पता नहीं, आई डोंट नो। थैंक यू सर, थैंक यू सर, थैंक यू।
सर शी इज नॉट देयर, वो बाद में देगी सर, उसकी तबीयत ठीक नहीं है इसलिए।
Yes, sir. Good evening, sir. My name is Monali More. My roll number is 227320. My topic is identifying gene using heterologous probes and oligonucleotide probes. First is introduction. A probe is a nucleic acid molecule with a strong affinity with a specific target. Probe and target base sequence must be complementary to each other, but depending on condition, then they do not necessarily have to the exactly complementary. Gene probe are used in a various blotting and in situ technique for the dictation of nucleic acid sequence. First is identifying specific DNA sequence in a library using heterologous probes. A probe that contains DNA which encoded for the gene of the interest but from another species is called a heterologous probe. cDNA probes can be used to identify and isolate specific gene, and a large number of genes have been cloned from both prokaryotic and eukaryotic in this way. It is also possible to identify specific gene in a genomic library by using a clone of similar gene from other organism as probes. For example,
mouse probes could be used in probe a human genomic library. Mouse genome is a 98% similarity with the human genome. As we know, maximum experiment was done on mouse, so the sequence of the DNA of particular gene is known. Making a probe using mouse DNA sequencing, having a radioisotope label, and then human genome degrade or denature. Then incubate with the probe from the mouse. It bind to the single stranded DNA it human genome because it is a complement to it. Such probes are called heterologous probes, and their effectiveness depending on a good degree of homologous between the probe and the gene. For the reason, the greatest success with this approach has come with highly conserved gene or with probes more specifically closely related to the organism from which this particular gene is to be isolated. Next is the identify gene or cDNA in library using oligonucleotide probes. Oligonucleotide probes are shorted straight a single stranded DNA or RNA used to detect the presence of complementary nucleotide acid sequence by a hybridization. A number of gene by a number of gene have been isolated from libraries by using synthetically made oligonucleotide probes. In this method, at least some of the amino acid sequence must be known for the protein encoded by the gene. In this case, it may be possible that consensus sequence can be determined from previously cloned version of the gene that is available in a gene bank, a computer database where a sequence are deposited and made available to research worldwide. Then because the uh, genetic code is a universal. Oligonucleotide about 20 nucleotides long can be designed that the translator it translated will give the known amino acid sequence. Because the degeneracy of the genetic code up to six different code can be specified given amino acid. A number of different oligonucleotides are made, all of the which could encoded the target amino acid sequence. These probes are known as gacimers. These mixed oligonucleotides are labeled and used to as probes to search the libraries with the hope that at least one of the oligonucleotide will dictate the gene or cDNA interest. If the probe is labeled radioactively, dictation is by audio radiography, whereas this probe is labeled non-radioactively. Dictation is done a colorimetry or with chemiluminescence. Thought not successfully all of the time. Oligonucleotide-based library screening has been extremely fruitful and has been allowed many genes that were missing molecular information to be cloned. Thank you. Awesome. Shorted nucleotide sequence. Probes that contain DNA which encoded for gene of the interest but from another species is known as heterologous probes. Yes. Um, this is chemical synthesis. पता नहीं sir. Okay sir.
Yes, sir. Yes, sir. Hello, sir. It is visible. Okay. Good evening, sir. Good evening, everyone. I am Miss Akanksha Navnath Nakwa, and my roll number is 227321. My topic name is Southern blot analysis of sequence of genome. So, what is introduction? Basically, what is a southern blotting? A southern name has been derived from in 1975. Sir Edwin Mellor Southern developed this technique that is widely used to detect fragments of DNA. Uh, Blotting means generally term for the transfer of macromolecules like DNA, RNA, and proteins. Uh, the southern blotting is defined as the whole process of separating DNA fragments by agarosal electrophoresis, transferring the fragments onto a filter, and hybridizing them with a labeled complementary probe is called southern blot analysis or southern blotting. So basically, southern blot is the molecular biological technique used for the detection of specific DNA sequence in the genomic DNA. Here is a principle. The key to this method is hybridization. Hybridization means it is a process of forming a double-stranded DNA molecule between a single-stranded DNA probe and a single-stranded target probe. Uh, it means single-stranded probe and a single-stranded DNA sequence binds together to form double-stranded DNA is called hybridization. There are two important features of hybridization. The reactions are specific. The probe will only bind to target with a complementary sequence. Separation of DNA fragments by gel electrophoresis and identified by labeled probe hybridization. So here is the procedure. Genomic DNA is first isolated, extracted, and purified from the cell. Sample of genomic DNA are cut with different restriction enzymes, each of which produces DNA fragments of different length depending on the location of the restriction sites. Then we perform agarose-gel electrophoresis. In agarose-gel electrophoresis, we load a sample of genomic DNA. The DNA restriction fragments are separated on the basis of molecular weight. After electrophoresis, the DNA is stained with ethidium bromide so that it can be seen under ultraviolet light. As DNA is double-stranded DNA, to perform the southern blotting, the double-stranded DNA have to be converted into single-stranded DNA. To convert double-stranded DNA into single-stranded, we treat the gel with mild alkali solution like NaOH. When the gel is dipped in the NaOH solution for some time, the double-stranded DNA in gel is converted to single-stranded DNA as the denaturation of hydrogen bonds takes place. Uh, the double-stranded DNA fragments are transferred to a membrane filter. A membrane fil- uh, there is a special membrane filter used in this technique, that is nitrocellulose membrane. First, we take a tray containing buffer solution. Then we place the gel in which the single-stranded DNA are present. Then we use the membrane. Uh, One thing here we have to remember that the size of the membrane filter should be equal to the size of gel. Then we place the Whatman paper or paper towel, and above the paper towel, we give some weight so that the paper towel and membrane will be pressed to the gel. After that, the buffer solution in tray, by capillary action, it moves towards upward direction. When this buffer reach to the gel, then the DNA present on gel will move towards the membrane. Along with the buffer, then the DNA will attach to the membrane filter. When paper towel becomes wet, that means our DNA is almost transferred to the filter paper. Then we remove the membrane filter. The fragments on the filter are arranged in exactly the same way as they were in the gel. After that, the membrane is incubated in the probe solution. Probes are attached to the detector molecule. Uh, Detector molecule will maybe radiolabel atom or fluorescent molecule. As probe is attached to the detector molecule, it is complementary sequence of a gene of interest sequence. On the basis of detector molecule, we visualize the membrane. Suppose the detector is radiolabeled on the probe, then the radiolab if the detector is radiolabeled or the probe is radiolabeled probe, then we perform autoradiography. By following these steps, we perform the Southern blot technique to detect the specific DNA sequence. Then here are some applications to identify specific DNA in a DNA sample. Southern blots are used in gene discovery, mapping, evolution and development studies, diagnostics and forensics. Used in prognosis of cancer and in prenatal diagnosis of genetic diseases. Southern blotting is also used in DNA fingerprinting, including maternity and paternity testing, criminal identification, forensic science, and also in personal identification. Here are advantages. Effective way to detect a specific DNA sequence in a large, complex sample of DNA. Second, can be used to quantify the amount of the present DNA in the sample. Increased sensitivity of fragments detection because of probes label used for amplifying signals. It is cheaper than the DNA sequencing. Here are some disadvantages. Southern blot requires large amount of DNA. This technique is not applicable in routine diagnostic setting. Southern blot is a time consuming. Here are some references from which I have presented my presentation, and thank you. Yes, sir. So there are three blotting techniques. Southern blotting is used for the DNA sequencing, and for RNA we use Western blotting, and for protein we use uh, Northern blotting. Sir, for RNA we use Northern blotting, and for protein we use Western blotting. Sir, because of paper towels, uh, because of paper towels, the uh, the pressure will uh, sir, because of paper towel, uh, the DNA will attach to the membrane filter. Means that print will come on the membrane filter. Uh, Probes. Sir, sir, DNA probe. Sir, first we will keep a smaller length then bigger. Thank you, sir. Thank you, sir.
Yes, sir. Sir, is it visible? Yeah, okay. Good evening. Myself, Khushali Parkhe. My roll number is 227322. My topic of presentation is Northern blot analysis of RNA. We'll start with the introduction. The Northern blot or RNA blot is a technique used in molecular biology research to study gene expression by detection of RNA or isolated mRNA in a sample. With Northern blotting, it is possible to observe cellular control over structure and function by determining the particular gene expression rates during differentiation and morphogenesis, as well as in abnormal or diseased conditions. Northern blotting involves the use of electrophoresis to separate the RNA samples by size and detection with a hybridization probe complementary to a part of or the entire target sequence. The Northern blot technique was developed in 1977 by James Alvine, David Kemp, and George Stark at Stanford University. The Northern blotting takes its name from its similarity to the first Northern blotting technique, the Southern blot, named after biologist Edwin Southern. The major difference is that RNA rather than DNA is analyzed in the Northern blot. Northern blotting involves the use of electrophoresis to separate RNA samples by size and detection with a hybridization probe complementary to part of or the entire target sequence. The RNA samples are most commonly separated on agarose gels containing formaldehyde as a denaturing agent for the RNA to limit secondary structure. The gels can be stained with ethidium bromide and viewed under the UV light to observe the quality and quantity of RNA before blotting. Here is the whole procedure. First, RNA are extracted from tissue sample and they are run in gel electrophoresis where they are separated according to their size. Then nitrocellulose membrane is placed on top of gel. On that membrane, blotting paper and weight are placed. Then through this process, the RNA move towards the membrane filter by capillary action. Here, the transfer buffer used for the blotting usually contains formamide. Then a label probe is hybridized with it. Then it is washed to ensure that probe are bound specifically to it, and the unbound are washed off. Then these hybrid bands are detected by autoradiograph. Applications are the Northern blots are particularly useful for determining the conditions under which specific genes are being expressed, including which tissues in a complex organism express which of its genes at the mRNA level. Detection of specific mRNA in a sample used in screening of recombinants by detecting the mRNA produced by the transgene in disease diagnosis, in gene expression studies, and in RNA splicing studies. It has its advantages as well as disadvantages. Its advantages are the strength of this method is its simplicity, specificity is relatively high. The quality and quantity of RNA can be measured on the gel prior to blotting. mRNA transcript is can be transcript size can be detected. Its disadvantages are time-consuming procedure. RNA samples can be degraded by RNases. Use of radioactive probes. These are the reference links. Thank you. Yes. So we can use RNA or DNA probes. Specifically, DNA probes are used. Uh, um, is there a stable stain? We can stain with ethidium bromide so that it will be visible by in under UV light. Other by chemiluminescence. Okay.
No, sir, she is not there. Yes, sir. Yes, sir. Yes, sir. It is visible? Yes, sir. Good afternoon, everyone. Myself Qureshi Farhanas, and my roll number is 227324. And my presentation topic is PCR, that is polymerase chain reaction, its principle and application. So let's start with what is PCR. So PCR is a polymerase chain reaction. It is a technique used in molecular biology to create several copies of a certain DNA segments, or it is a laboratory technique used to amplify DNA sequences. So this technique was developed in 1983 by Kerry Mullis. He was an American biochemist. PCR has made it possible to generate millions of copies of a small segment of DNA. This tool was commonly used in the molecular biology and biotechnology laboratory. So next is principle of PCR. The PCR is the polymerase chain reaction is to make a huge number of copies of a gene. This is necessary to have enough starting template for sequencing. There are three major steps in PCR which are repeated for 30 or 40 cycles. This is done on an automated cycler which can heat and cool the tubes with a reaction mixture in a very short time. That automated cycler is also known as a uh, thermocycler. So there are three steps. First is denaturation, then annealing and extension. So denaturation occurs when the reaction mixture is heated to 94 degrees Celsius for about 0.5 to 2 minutes. This breaks the hydrogen bonds between the two strands of DNA and converts it into a single stranded DNA. This single stranded now act as a template for the production of new strands of DNA. The temperature should be provided for a longer time to ensure that the separation of two strands DNA uh, remains. Then the next step is annealing. This reaction temperature is lowered to 54 to 60 degrees Celsius for around 20 to 40 seconds. Here, the primers bind to their complementary sequences on the template DNA. Primers are single strand sequence of DNA or RNA around 20 to 40 20 to 30 bases in long. They serve as a starting point for the synthesis of DNA. Uh, the two separated strands run in the opposite directions, and consequently there are two primers, a forward primer and a reverse primer. So the third last step is extension. Extension. At this step, the temperature is raised to 70 to 80 degrees Celsius. The bases are added to the 3' end of the primer by the Taq polymerase enzyme. This extension, the DNA in the 5' to 3' direction. The DNA polymerase adds about 1,000 base pair per minute under optimum conditions. Taq polymerase can tolerate very high temperatures. It attaches to the primer and adds DNA bases to the single strand. As a result, uh, a double-stranded DNA molecule is obtained. These three steps are repeated 20 to 40 times in order to obtain a number of sequence of DNA interest in a very short time of period. So this is the steps involved in PCR. First, denaturation, annealing, and the third is extension. So next are the applications of PCR. There are many applications in PCR. In PCR, uh, so let's start with DNA fingerprinting. DNA fingerprinting printing, it is used in forensics. So in forensics, PCR can be used for DNA, for example, to amplify trace amounts of DNA in samples such as hair, blood, or semen collected from a crime scene. The amplified DNA can be analyzed and compared with DNA from victim and suspect. The result can be used to implicate or exonerate suspects in the crime. This analysis called DNA typing, DNA fingerprinting, or DNA profiling. Hmm. The next one is prenatal genetic disease. So first, what is genetic diseases? Genetic diseases are inherited medical conditions caused by DNA abnormality. Disease Disease are caused by mutation that are inherited from the parents and are present in an individual at birth is called genetic disease. So, for example, in a family, a genetic disease is inherited since a long time. So, consider if a woman is expecting a baby and she is scared about the genetic disease which she has in her family and can occur to baby or infant. So in this case, we can use a PCR technique by obtaining her fetus DNA through amniotic fluid or bloodstream of a mother. So after obtaining a DNA, we will run a PCR using a specific primer known as disease-specific primers. If PCR get amplified, then the particular disease uh, is present in the baby, or if PCR do not get amplified, then the genetic disease is not present in the fetus or a baby. So next is sex determination. Sex determination. So polymerase chain reaction is used to identify the sex of any individual. For example, we have a biological sample and we don't know the sex of the sample. So we extract the DNA from a given biological sample, and then will run PCR using a specific primers which helps to determine the sex of the individual. So these are the sex specific primers. After then we will run PCR. After PCR is completed, we check we will run the age agarose gel electrophoresis. If we see double band on a gel, then it would be male sample, and if a single band is seen, then it will be a female sample, because male has XY chromosomes, and so we get double bands, and female has XX chromosome, so we get a single band. So next is quantitative PCR. Quantitative PCR or qPCR is also called as real-time PCR or quantitative real-time PCR. It is a PCR-based technique that couples amplification of a target DNA sequence with quantification of the concentration of that DNA species in the reaction. Here in quantitative PCR, we get amplified DNA, but we can also find the concentration of the DNA species. So quantitative reverse transcription involves the detection and quantification of RNA. The next is diagnosis of infectious diseases. In diagnosis, in diagnosis of infection and infectious diseases, for example, it is used to diagnose infection by the viruses. We take blood of a patient that has been infected by the virus. Then we amplify the DNA of the virus so that we are able to study which type of virus it is and its properties. So next is organ transplantation. In uh, organ transplantation is a medical procedure in which an organ is removed from from one body and placed in the another body of the recipient to replace a damaged or a missing organ. In organ transplantation, polymerase chain reaction plays a role to detect the correct and suitable organ for an individual. Before organ transplantation, it is very necessary to check the graft acceptance and graft rejection. So, uh, for example, if a person is a donor and B person is a recipient or receptor. So if A person is donated his or her organ to a B person, and that B person accepting the organ will show strong immune response. So to avoid this, we use HLA typing PCR, that is uh, antigen human leukocyte antigen polymerase chain reaction. In this, we use HLA-LSP, that is human leukocyte antigen locus specific primer, because human genome contains HLA and HLA is highly variable. It means that the A and B person will have different genome HLA contained. In this case, we'll collect both A and B person's DNA and will run a PCR separately. It will be HLA typing PCR, and we will use the HLA-SP specific primer. In If in case of A person, the PCR will get amplify, and in case of B person, the PCR will not get amplified, then it means body is not ready for organ transplantation. That means graft rejection. And if A and B persons both get amplified, then it means body is ready for transplantation, and it means HLA locus is present in both body or individual. The body is ready for organ transplantation. So these are the references. Thank you, sir. Thank you, sir. Yes, sir. Yes, sir. Taq polymerase, Pyrococcus furiosus, that is PFU polymerase, and Taq polymerase is Thermus aquaticus, and Thermococcus litoralis, that is TLI, or also known as vent polymerase. Yes, sir. Hmm. After how many cycles? 10 cycles. Sir, please repeat the question. Hmm. हाँ. 1 raised to 4, no? 1 raised to 4 raised to n. 1 divided by 4 raised to n, no, sir? Yeah, 2 raised to n. No, 2 raised to n. 2 raised to n, no, sir? Yes, sir. Re- एनिलिंग में तो सर जो दो मतलब एक स्ट्रैंड होता है वो प्राइमर्स बाइंड होते हैं एनिलिंग में डोंट नो थैंक यू सर
Yes, yes, sir. Yes, sir. Okay, sir. Is it visible, sir? Okay. Good evening, sir. Good evening, everyone. Myself, Devraj Sharad Sappa. My roll number is 227325, and my topic is contributive PCR. Uh, so I will like to start with PCR. Though my friend Farhanas has explained it very well, still I will like to brush up a little. So PCR is nothing but polymerase chain reaction. Polymerase chain reaction or PCR is a commonly used laboratory technique that was invented in 1980s by Carey Mullis. The method has many applications in different fields, ranging from identifying individuals, forensic science, detecting pathogens in water supply, and genetic testing in medicine. PCR works by first obtaining a sample that is contains genetic information, DNA, such as blood, then using a specialized enzyme called DNA polymerase. Researchers can amplify and make billions of copies of a specific segment within a DNA that they are interested in. So for any PCR setup, we required. So this is first diagram is PCR machine. For any PCR setup, we require certain equipments. So this is like tips, micropipettes, and this is a kind of tube. And we also required some for reaction mix. We require some of the like nucleotides, primer, template, enzyme. We require to process the DNA cycle. So we mix all the that requirement in a F-and-F tube with the help of micropipette and tubes and tips. So this is the mechanism of PCR. They are divided in three steps: denaturation, annealing, and extension. So in the denaturation, high temperature incubation is used to melt or separate the double-stranded DNA into single strand, and uh, at a 95 degrees Celsius. And in annealing, primers, specific primers are attached to the DNA strand, and co- complementary sequence have an opportunity to hybridize. So an appropriate temperature is used that is based on the calculated melting temperature. That is 5 degrees Celsius below the primer. And next one is extension. So tax polymerase builds a complementary stand with the help of primers and complete the stand. And now these two stands can make proliferate into another four, five, like that cycles continues. So we will. There are different types of PCR, like conventional PCR, multiplex PCR, RT-PCR, quantitative PCR, methylation specific PCR, assembly PCR, and colony PCR. So, uh, my topic is quantitative PCR. So, quantitative PCR or qPCR, also known as real-time PCR or quantitative real-time PCR, is a PCR-based technique that couples amplification of of a concentration uh, that DNA species in the reaction. Both PCR and quantitative PCR are polymerase chain reaction techniques used to amplify specific section of DNA. The main difference between the two is that the quantitative PCR is a real-time method while the PCR is not. This means that with quantitative PCR, you can monitor the amplification of your targeted DNA in real time. It is happening. So principle of quantitative PCR is this the same principle of amplification of PCR is employed in the real-time PCR. But instead of looking at bands on the gel at the end of reaction, the process is monitored in the real time. The reaction is placed into a real-time PCR machine that watches the reaction occur with the camera or detector. Although many different techniques are used to monitor the progress of PCR reaction, all have one thing in common, that they are all linked with the amplification of the DNA to the generation of fluorescence, which can simply be detected with a camera during each PCR cycle. Hence, as the number of gene copies increases during the reaction, so does the fluorescence indicating the progress of the reactions. So, these are some steps of quantitative PCR. Quantitative PCR steps are there are two steps in which quantitative PCR works. First is amplification, and second is detection. So in amplification, same as I said in PCR also, three different stages are happened: denaturation, annealing, and and extensions. As you can see here. And detection is based on fluorescence technology. So different fluorescent dyes are used. The different markers are used in real-time quantitative PCR, like cyber green. That we it is a dye that emits promit fluorescence signal when it binds to the minor groove of DNA. Other fluorescents like ethylene bromide or arginine orange can also be used to, but cyber green is also better for used for the high signal intensity. So as you can see, it also has three stages: denaturation, polymerization, and signal detection. So in denature, this fluorescence dye break get attached to the double-stranded DNA and hybrid and then in polymerization it start with the primer to go along with the segment. Another is a tachman. So tachman is a probe. It is hybridizes probe which bear a reporter dye at the far end prime. Under the normal condition, the probe remain coiled on itself, bringing the fluorescence dye near the quencher, which inhibits or quenches of fluorescent signal of the dye. The oligonucleotide of the Taq polymerase has a homologous region with the target gene, and thus, when the target sequence is present in a mixture, it binds with the sample of the DNA. So these are some fluorescent markers used in quantitative PCRs, like fluorescent dye-based real-time PCR is nothing but cyber green, and DNA probe that I just explained is Tachman. So this is like a second step of quantitative PCR, which is detection. So detection is based on the fluorescence technology. The specimen is first kept in proper well, like you can see this 69 well plate, uh, and subjected to the thermal cycle like normal PCR. And the machine, however, in the real-time PCR is subjected to tungsten or halogen source. As you can see here, tungsten and halogen lamp is there. That leads to the fluorescent marker and added to the sample, and signal is amplified with the amplification of copy number of sample of DNA. The emitted signal is detected by a detector that is connected on the software computer after conversion into digital signal, and then we can see the graph or any results. So there are so many advantages of quantitative PCR. Like it has many. It gives a look into the reaction. What is the reaction that is help to decide which reaction have worked well and which have failed. The efficiency of the reaction can be precisely calculated. There is no need to run the PCR product out on a gel after the reaction as the melt curve analysis serve the purpose. The real-time PCR data can be used to perform truly quantitative analysis of the gene expression. In comparison, old-fashioned PCR was only ever semi-quantitative at the best. It's faster than the normal PCR, less complexity at the quantitative quantification of the sample. So there are so many applications like gene expression analysis in cancer research, drug research, disease diagnosis and management, like HIV, AIDS, viral quantification, in is virus is present in the sample or not, in how much quantity virus is present, genetically modified food, animal and plant breeding, gene copy number. These are some references which I took, sir, and I also prefer iGenetics for it. Thank you. mRNA. Yes, sir. Yes, yes. TT. Uh, sorry, sir. DNA polymerase, E. coli, sir. Yes, thank you, sir.
Yes, sir. Ah. Sir, it is visible. So myself Nisha Sanjay Sarate, roll number 227326. My topic is DNA cloning basic steps. DNA cloning. DNA cloning is the process of making multiple copies of particular segment of DNA. During this technique, the selected DNA fragment is inserted into plasmid. Using a enzyme restriction enzyme and DNA ligase are used in the process. The restriction enzymes are used to cut DNA fragments at specific sequence, and DNA ligase enzymes are used to join the nicks. The recombinant DNA thus is introduced into bacteria. These bacteria reproduce and produce an exact copy of plasmid. These copies are known as clones. So DNA cloning's basic steps. DNA cloning takes place in three types of steps: cutting and pasting DNA, bacteria transformation and selection, and third step is protein production. So first step is cutting and pasting DNA. Two types of enzymes are used in this method: restriction enzyme and DNA ligase. The restriction enzymes cut the DNA at specific target sequence. The target gene is inserted into the cut site and it's ligated by DNA ligase. This is known as recombinant plasmids. So plasmid specific target sequence in plasmid and target gene is inserted with DNA ligase. So here is recombinant plasmid. Bacterial transformation and selection. The recombinant plasmid is introduced into bacteria such as E. coli. The bacteria are subjected to very high temperatures, which compel to take up the DNA. This process is known as transformation. The plasmid contains an antibiotic resistant gene, which helps to them to survive in the presence of antibiotic. The plasmid containing bacteria are selected on nutrient containing antibiotics. The transformed bacteria survive while the ones without a plasmid die. Last step is protein production. The plasmid containing bacteria are cultured and the bacteria are provided with a chemical signal that helps to them target the protein. After protein production, the bacteria are split once upon to release it. The protein is purified and the target protein is isolated from other content of the cell. So here colony is grown in large culture. Then the culture are mixed in the flask. Bacteria are induced express gene. So here gene is transcribed into mRNA. mRNA is translated into protein. Example, insulin. Importance of DNA cloning. The DNA molecules produced through the cloning techniques are used for many purposes, which includes DNA cloning. DNA cloning can be used to make proteins such as insulin with biomedical techniques. It is used to develop recombinant version of the non-functional gene to understand the functioning of normal gene. This is applied in gene therapies also. It helps to analyze the effect of mutation on particular gene. So here reference and thank you, sir. DNA ligase enzyme used to ligate pasting of DNA and restriction enzyme is used to cut the DNA. The recombinant organism is the plasmid cutting and the target gene specific is ligated in the plasmid. So this is a recombinant organism or DNA. Thank you, sir.
Yes, sir. Yes, sir. Sir, is it visible? Mhm. I've shared, sir. Wait a minute, sir. Sir, is it visible now? Okay. So, good evening, sir. My name is Shreya More, and my topic is restriction enzymes. So, what is restriction enzyme? A restriction enzyme is a protein which is isolated from a bacteria that cleaves the DNA sequences at sequence specific sites, producing DNA fragments with a known sequence at each end. So, the restriction enzymes are also called as molecular scissors as they cleave the DNA near or the specific recognition sequences. And restriction enzyme was discovered by Werner Erber. So there are two different kinds of restriction enzymes. First is exonucleases and second is endonucleases. So exonucleases are nucleases which cut off nucleotides from 5' to 3' end of DNA molecule, and it also acts as proofreading or during DNA polymerization and DNA replication. So the example is exonuclease 1 and exonuclease 2. So what is endonucleases? So endonucleases are nucleases which cleave DNA. Uh, हाँ एक मिनट नहीं दिख रहा है दूसरा वाला स्लाइड. तेरा भी दिख रहा? There are two different kinds वाला वो वाला स्लाइड पे. हाँ. So what is endonucleases? So endonucleases are nucleases which cleave DNA at any point except the ends. So it can recognize specific base sequence within DNA or RNA molecule. So the examples are HIND 3 and BAMHI. So there are basically three types of restriction enzymes: type 1, type 2, and type 3. So type 1 restriction enzymes cut the DNA far from the recognition sequences. They are complex multi-subunit restriction and modification enzymes, and they cleavage occurs approximately 1,000 base pair away from the recognition site. So type 2, these enzymes cut at specific positions closer or within the restriction sites. They are also used for DNA analysis and gene cloning. So in this type 2 restriction enzyme, there are two different types of cuts basically, that is blunt end and sticky end. So in blunt end, uh, both strand we get of equal length, and in sticky end one is larger one and other is smaller, uh, unpair basis on either strand, and it is very more effective than the blunt ends. And type 3 restriction enzymes, these are the multifunctional proteins with two subunits, that is race and mod. So the race is used to is used for nuclease action, and mod is used for the recognition and modification of DNA. So this is some of the types with its sites and location. So type 1, it cleaves the basically away from 1000 base pair away from the recognition site, and its location is endonuclease, and methyl is located on a single protein molecule. Type 2, it cleaves the DNA within the specific recognition site. And type 3, it recognize randomly 24 to 26 base pair away from the recognition site. So this is so in this, as you can see, first the restriction endonuclease have non-specific binding with DNA, and after that this endonucleases then gets jumped over a long distance, so it recognizes its recognition restriction site. So this restriction endonucleases jump over and forms a specific DNA binding, specific binding with the DNA. After the binding, it cuts the DNA at specific binding site, and the product is released. So the applications are restriction enzymes are used in gene cloning and protein expression experiments. The restriction enzymes are also used in biotechnology as to cut the DNA into smaller strands, and it is also used in genetic engineering. So here are some advantages and disadvantages. So restriction enzymes have proved to be invaluable for the physical mapping of DNA, as they offer unparalleled opportunities for diagnosing DNA sequence content and are used in fields as criminal forensics and basic research. So the disadvantages are time. The enzyme concentration is low. So these are the references link. So it is basically an organic compound. %50. 4 is to N. No? So neosisomers are the in which the carbon atoms form two forming continuous chain. Yeah, so it's a peptide bond. फॉस्फोडायस्टर ना? हाँ नहीं बाहर गई है.
Yes, sir. Yes, sir. हाँ sir. 1 minute. Hello, sir. So good evening, guys. My name is Prathamesh Kavre, and my topic is plasmid explanation and properties of one plasmid. So first, what are plasmids? Plasmids are small circular DNA fragments, double-stranded, self-replicating extra chromosomal structure found in many microorganisms. So this is the structure of plasmid. So structure of plasmid, it includes origin of replication, selectable marker, promoter region, primer binding site, and multiple cloning sites. The size of plasmid varies from 2 kb to 200 kb. It is the extra chromosomal element of the cell which is not required for growth of development of the cell. Most of the plasmids contains the TRA gene, which is transferred gene and is essential in transferring the plasmid from one cell to another. So what is the type of plasmid? Based on presence of TRA gene, plasmids can be classified into two types: conjugative plasmids. These plasmids contain TRA gene and are commonly seen in bacteria, and non-conjugative plasmids. These types of plasmids lack the TRA gene. So what are the types of plasmids? Based on their functions, they classified into following types. First is F plasmid, fertility plasmid. They contain the TRA gene and hence can be transferred from one cell to another. R plasmid, resistance plasmid. These plasmid contain and transmit gene to antibiotic resistance from one cell to another. Cold plasmid, there are known as bacteriogenic plasmid because they produce bacteriocinogens. Degradative plasmid, this type of plasmids have the ability to digest unusual substances such as toluene, camphor, salicylic acid. Tiffany virulence plasmid, this plasmid produce virulence factor that enables the bacteria to infect other cells. Bacteria containing virulence plasmid are able to infect the plant, animal, and human cells. So, what is the property of plasmid? They are extra chromosomal DNA fragment present in cell. They are double-stranded structure. Expection are the linear plasmid in bacteria, Streptomyces species. They can replicate independently. Plasmids are also known as sex factor, conjugate, extra chromosomal replicon, or transfer factor. Thank you, sir. Yes, sir. सर नहीं मालूम सर दूसरा सर नहीं पता सर। नहीं पता सर।
सर मेरा प्रेजेंटेशन डाउनलोड नहीं हो रहा है, 1 मिनट प्लीज। ओके सर।